All right. So we've been on top of this mountain, and now we have to start coming down. And I want to finish with one little parting shot. On Wednesday, we talked about sequencing DNA, the way that we can not only duplicate any piece of DNA that we want, but we can read any piece of DNA that we want, thanks to the polymerase chain reaction as worked out by originally by Kary Mullis and then refined quite a bit since then. Now, if you remember, we started genetics when I mentioned that there was a hole in Darwin's original theory of evolution by natural selection, and that was that he did not understand how heredity works, why and how organisms pass on their traits from one generation to the next. Now, he knew very well that it happened. You can see that in any family, but why it happened, what was causing it, was a big black box. Now, we've opened that black box, I think, pretty well, about as well as can be expected in the time that we've got. And we've got a much more complete understanding of how heredity operates than Darwin ever did. It's also worth noting that not only do we understand heredity a whole lot better, the ability to read DNA provides whole new lines of evidence for reconstructing evolutionary history. We can do it using DNA sequences as evidence for testing hypotheses. To give you a tiny bit of the flavor, these happen to be short snips of actual DNA. I believe it is the DNA gene that codes for one of the RNAs that is a structural part of ribosomes. On the top, you've got hippo DNA, and below, you've got DNA from 3 different whales. The orca, or quote, killer whale, sperm whale, and blue whale, the largest animal of all. And if you put them up next to each other, you can see that they are the same in a lot of places, but there are some differences. So if we draw red boxes around each one of those differences, you can see that there are, well, you can see where they are. And you can see that, here, I'll do this in different colors. Some of these nucleotides are the same in all 3 whales, but they're not found in the hippo. So at position number 5 in its DNA, the hippo's got a cytosine, a C, where all 3 whales have an adenine, an A. And then over here at this position, the hippo's got a G, where all 3 whales have an A. And here the hippo's got an A, where all 3 whales have a C. And then you can see there are some other patterns within here, where you might have 2 whales that share a nucleotide that the other whale and the hippo do not. So right here, the orca and the sperm whale both have an A, whereas the hippo and the blue whale both have a G. And there's a spot where the orca and the sperm whale have a C, where the hippo and blue whale have a T. But here's a place where the sperm and blue whale have a T, where the hippo and the orca have a C, and so on. And what you can do is take these similarities and use various methods that we don't really have time to get into, but they're very easy to implement if you know how to code. They're pretty easy for a computer to handle. And we can find patterns in these similarities and differences that we see in DNA. These differences result from mutations in the past. And if 2 different species split from a common ancestor very recently, there probably has not been very much time for mutations to happen. On the other hand, if their last common ancestor was a very long time ago, they will probably have more differences because there's been more time for mutations to occur. And you can use that fact, and you can use various other features of similarities to come up with hypotheses for how groups of living things have evolved. And this is a fairly recent result using computer analysis methods on very large sets of DNA data. You can see down here, the baleen whales right here include blue whales and humpbacks and things like that, that don't have teeth, but feed with filtering plates in the mouth called baleen. Toothed whales right here include orcas. They also include dolphins and sperm whales. And based on similarities in their DNA, we don't really need to get into what those are. They're listed down here. But we think that toothed whales and baleen whales shared a common ancestor. And both toothed whales and baleen whales shared a common ancestor with hippos way back here. And whales plus hippos share a common ancestor with chevrotains and pecorans, which is basically almost all of the hoofed mammals. And if you go even farther back in time, you'd find common ancestry with pigs and even with camels. And the data that we're using to support this hypothesis that we've drawn is DNA-based data. We can find these patterns in the similarities and differences in the DNA of different groups, and we can use those to draw cladograms, just as we could use similarities in physical features to draw cladograms, like we talked about way back around week 5. And in this case, there happens to be a fossil record. We don't always have relevant fossils. There are some groups where the fossil record is quite incomplete. But where we have it, we can use DNA and fossils as sources of data to test hypotheses that we've drawn up. So for example, this is a fossil from Pakistan, roughly 46 million years old. It is very completely known. It's called Ambulocetus, and it's got features in common with whales. For one thing, we're pretty sure it swam. For another thing, the teeth look like early toothed whale teeth. For another thing, the structure of the inner ear suggests that it could dive pretty deeply. And yet it's still got 4 legs. Although how well it was able to walk is not entirely sure. It may have been able to kind of, hump along on the beach, but it spent most of its time in water. And it shares some features with the ancestors of later hoofed mammals and others with whales. This may not be the common ancestor of all whales, but it's very close to it. And in this case, it pretty well confirms our DNA-based hypothesis that whales evolved from a group of land mammals that shared features with the hoofed mammals that came after them. So we can cross-check evolutionary hypotheses, creating one based on one source of data and then testing it against information from another source of data. So I just wanted to kind of tie that up and kind of bring us back to where we started this whole exploration of genetics. Ta-da! Incidentally, we can also use this to track the evolution of virus strains. Viruses tend to have a very high mutation rate. And we've seen this play out over the past 2 years. These new strains of COVID, Alpha, Beta, Delta, Omicron, now Omicron B2, Omicron's little brother. All of these appeared by mutation and persisted because of natural selection. And the reason why you keep needing new flu shots and the reason why you keep needing COVID boosters and The reason why new strains keep appearing is just plain old mutation plus natural selection. And we can use DNA sequencing to track the process. So this may not be up to date even more, but right there is the COVID that we've been grappling with for the past 2 years. And it shares similarities with various animal viruses. Down here, I think this is SARS. That was a real problem in China back in, I want to say, about 2015. That's another one. And that has relationships to various other viruses. And yeah, we can basically just keep tracing in almost real time how viruses are evolving using evidence from DNA strains. And we do, in fact, do this on a regular basis. This is done pretty much routinely now. Okay, actually, I take that back. The first thing that I circled was MERS. That was Middle East Respiratory Syndrome, which was a COVID-like illness that we had an outbreak of about 10 years ago. SARS right there, we had an outbreak of in China. And then the actual COVID is down here. So you can see that all of these viruses are related, and they've jumped to humans several times from various viruses that affected animals. And we can do even better. Even if we don't have fossils, we can explore the evolution of all life on Earth. And if you could see all of it, it would look sort of like this. This is a cladogram. It's just been bent into a circle. And metazoa right there, that's basically all animals, vertebrates and invertebrates. And yeah, humanity is one of these little branches up here in the left corner. I'm not even sure which one it is. That could be us. Over here, all of these little branches right there, those are different fungi, plants, and green algae are over here. These are all various groups of protists. And then we have bacteria here, archaea, and everything going back to a single common ancestor that we don't have direct fossils of, but it must have existed at one time. It's affectionately known as LUCA, the last universal common ancestor. And we know this because even bacteria and archaea have much deeper genetic similarities with, quote, advanced life forms, including you, than we realized until we could sequence DNA. You have more in common with bacteria than you might be comfortable thinking about. And we can see some of that if we start digging down and looking at you at the cellular level. So way back when, I talked about how living things are made up of cells. We talked about how some groups have cell walls and others don't. Eukaryotes have nuclei and prokaryotes don't. There's a lot more complexity in cells that we didn't go over back around, week 2 and 3, but we can get to it now that we've had a good shakedown in, talking about genetics. Every cell, prokaryote, eukaryote, doesn't matter, as long as it's alive, contains a flowing substance called cytoplasm. Now, cytoplasm is mostly water. There's dissolved salts in cytoplasm. There can be a lot of free-floating proteins and various other things like that suspended in cytoplasm. And everything that's inside the cell floats in the cytoplasm or is suspended in it. What you're looking at here, this big blob, is a big protist. It's a single-celled critter called an amoeba. The nucleus is right there. There's a big empty space right here that's filled with wastewater that the amoeba is going to expel. That's called a vacuole. And you can see various things suspended in the cytoplasm as the amoeba moves by kind of flowing along. And the amoeba is about to engulf this green thing at the right. That's another protist. It's called Paramecium bursaria, and the amoeba is about to eat it. And the amoeba moves and gets around because it's able to make its own cytoplasm flow. What surrounds the cytoplasm is a membrane, very flexible. It's called the plasma membrane. And the plasma membrane is made up of lipids. Now, I know you saw some of these in the lab where you looked at food chemistry. Lipids include fats and oils. They also include waxes, incidentally. Your earwax is a lipid. You might remember that lipids turn black or brown when you expose them to Sudan-4. The question's been asked, does cytoplasm contain all the nutrients in the cell, or is there another organelle that does that? Answer, it depends on what kind of cell it is. Some cell types might contain special storage structures for nutrients. Plant cells, for example, may have special granules inside the cell that contain starch. There's probably always some nutrient floating around in the cytoplasm. Plant cells would have specialized starch granules. The cells in your fatty tissue contain basically big bubbles of fat, but you don't see that in other cells. So the answer is it depends on what kind of cell you're looking at. Anyhow, you don't need to learn all of these terms that are shown here on the diagram, but molecules of the type of lipids that make up a cell membrane, each molecule is shaped like it's got a head and two tails. And when lipids form a membrane, the heads all stick to the outside and the tails all point inward. So you have what looks like kind of a sandwich, except it's made up of lipid molecules. And there can be proteins that are embedded in the membrane, and they might stick out of one side, they might stick out of the other. Some of those proteins are shaped like little tubes and enable certain nutrients to go in, like this right there. That transport protein has a little hole in it, and certain substances can pass in, but not others. Some proteins stick outside and go inside. These are ways in which the cell may be able to respond to certain things. There's lots of them. We don't need to go into all of them, but these proteins allow cells to transport molecules in or sometimes out, respond to outside stimuli, things like that. And that plasma membrane isn't really a solid. It's not like a balloon made out of rubber. It's more like a film of, well, it is a very thin film of what's technically a liquid. If you've ever blown soap bubbles on a nice summer day, and you've watched the way that light shines through a soap bubble, you may have been able to see that the liquid that makes up the bubble, the soap liquid, is flowing over the surface of the bubble. You can tell because you can see these colors kind of shift and writhe as as the bubble floats past, and that's caused by liquid flow. And a cell membrane is a thin liquid film. It's not really a solid. All of those proteins can move within that film of liquid that makes up the plasma membrane. And because cell membranes are films of liquid, it's easy for them to fuse together or pinch apart. And if you've ever really, this sounds kind of weird, have you ever taken a really good look at soap suds? Like you're washing dishes in the sink and you ever really look at soap suds? It sounds kind of weird, but what you'll see if you do is that adjacent bubbles sometimes fuse, right? You'll have two small bubbles next to each other, and then they'll merge and suddenly become one larger bubble. And it's also possible to make large bubbles break up into a bunch of smaller bubbles. This is what makes it possible for cells to divide. When a cell divides, it's not really tearing the cell membrane the way that you'd have to tear a balloon or something like that. That liquid plasma membrane is simply just pinches in two, the way that a soap bubble can be made to pinch in two. Okay, so all cells, you've got a cell membrane that separates the out from the in, and cytoplasm on the inside. As we talked about a long time ago, most prokaryotes have this cell wall outside of the cell membrane. So there's the cell wall outlined in red. And inside a prokaryote, there's a central region where the DNA is located. It's called the nucleoid. And it shows up here as, kind of looks like a spaghetti serving, just kind of jumbled up in the middle of the cell. But the nucleoid doesn't have a membrane surrounding it. The DNA is just floating in the middle as one big clump, but it's not separated from the rest of the cell. Everything is happening within that cell, within that one cell wall and cell membrane. These black specks outside the nucleoid, many of those are going to be ribosomes, and they're just floating around freely. So not a lot of detail inside of a prokaryote. Bacterial DNA happens to be organized differently from eukaryote DNA. Eukaryotes have chromosomes that are long strips of DNA. Each chromosome is one long piece with a beginning and an end. Bacterial DNA is organized into one giant loop. If you could take that nucleoid DNA and somehow untangle it, you'd get one enormous loop of DNA. And bacteria also have a number of smaller loops that are called plasmids. This is a transmission electron micrograph of a couple of isolated bacterial plasmids. Each of those is one DNA molecule, each of those two little tangled loops. And bacteria can actually swap plasmids. When bacteria have their version of sex, they will pair up, and one of the bacteria will pass a plasmid to the other. Bacteria can also pick up plasmids from the environment. And this is what was going on when Griffith did his experiment that demonstrated the transforming principle. Those living bacteria were picking up plasmids that had been released by the dead bacteria. They were picking up these loops of DNA that bacteria can pass around. Bacteria can swap plasmids I don't know, like maybe you used to swap Pokemon cards or something like that. Or maybe it was Yu-Gi-Oh cards in your day. I don't know. Eukaryotes don't have this. Eukaryote DNA is linear. It's strips, not loops. And nucleus have that defined nucleus. And I seem to have misspelled it mucleus, but it's nucleus with an N as in November. And the nucleus is bounded by a double layer of membrane called the nuclear membrane, which makes sense. What you're looking at here is a number of eukaryotic cells that have been stained with fluorescent stains and imaged through a special microscope that can pick up this fluorescence. And the nuclei are shown in blue. I think that stain reacts with DNA specifically. And you can see that the blue lumps right there form these nice tight little packages. That's the nucleus of each one of these cells. And what separates the nucleus from the rest of the cell is the nuclear membrane, which you might just barely be able to pick up in red. That happens to be a cross-section through a plant cell. And I'm outlining the nuclear membrane right here. The cell is not actively dividing, so all of the grainy stuff inside the nuclear membrane, that's the chromosomes, that's those strips of DNA. But since the cell is not dividing, those chromosomes are not packaged up, and they wouldn't be visible under the light microscope. And even under this microscope, you can't really see individual chromosomes. They're all uncoiled and jumbled together. Inside the nucleus, there's a dark spot, and you can often see this under a light microscope. This is called a nucleolus. And the nucleolus happens to be where ribosomes themselves are assembled. Cell has to make its own ribosomes, and it's done in the nucleolus. What you're looking at there is the surface of a nucleus. It's been imaged with a special type of microscopy called freeze fracture transmission electron microscopy. And the nucleus is this big round thing right here. And instead of looking at a slice through the nucleus like that last picture, you're looking at the surface of the nucleus, and you can see there's a bunch of little round specks right here, like pepperonis on a pizza. I'm circling a few. Those are the nuclear pores. And they're not just holes in the nucleus. There are complex proteins called porins that act as little gateways. So a nuclear pore can open and close, kind of like the blast door on the Death Star, if you remember that scene from Star Wars Episode 4, the thing that Harrison Ford runs through just before it gets shut. The porins look kind of like that. And they allow RNA to pass out of the nucleus and get out to the ribosomes. They allow ribosomes that are made in the nucleus to pass out to the cytoplasm. They may allow certain proteins to get in. So they regulate the traffic coming into and out of the nucleus. They're the gates that allow some molecules, but not others, to enter or leave. So what's in the cytoplasm? We may not get to all of this today, but I'll cover as much of it as I can. A lot of the cytoplasm is not just fluid. It's filled with structures that collectively make up what we call the endomembrane system. A lot of folds and sacs of membrane that play a key role in the cell's metabolism. And probably the largest part is a network of folds and tubes of membrane. It's the same kind of membrane as the plasma membrane, but it's in the cell, not on the outside of it. And it functions as kind of a transport network within the cell, and it's called the ER, which is short for endoplasmic reticulum. Reticulum's Latin for a little net. So this is the little network. Endo means inside, and then plasmic refers to the cytoplasm. And again, if you don't care to say endoplasmic reticulum, you can just call it the ER. This is important because a lot of cells make proteins, and in many cases, those proteins have to be exported. You've got cells that make, for example, all of the enzymes that you use to digest food. Those enzymes are made in the cell, but they have to be exported out of the cell in order to get into your saliva or into your stomach or intestine or wherever they need to go to do what they're supposed to do. So the ER is where proteins and other products that are exported are made and transferred around. They'll go from the ER to a body inside the cell called the Golgi body. The Golgi body is named after an Italian who discovered it back in, he shared the Nobel Prize for his discovery back in 1905, a guy named Camillo Golgi. And you could think of the Golgi body as being the packaging and shipping center. A lot of things get made on the ER. The Golgi is where a lot of products are packaged and shipped out. And in addition to the Golgi, there also may be many little sacs of membrane inside a eukaryotic cell that can be small or big. If they're big, they're often called vacuoles. If they're very small, they're usually called vesicles. And that's what a slice through the endoplasmic reticulum looks like. You're not really seeing it in 3D. You're seeing a cut through it, imaged on a device called a transmission electron microscope. And what you're looking at is, imagine taking a folded sheet, like a bed sheet. Imagine folding it on itself several times and then cutting a slice through it. You'd see a whole bunch of layers of fabric. Well, here you're seeing a whole bunch of layers of membrane that make up this very complex folded structure. And incidentally, among the folds, you can see that in some areas, you've got lots of little black specks. I'm trying to circle a few, but they're not very big. They're about the size of pepper grains on my screen. Those are ribosomes. And in cells that make lots of protein for export, the ER is studded with ribosomes, and it's called rough ER. It looks kind of rough, kind of like maybe strips of sandpaper under the transmission electron microscope, but instead of grains of sand, it's studded with millions of ribosomes right there. Cells might also have ER that is not studded with ribosomes, and we call that smooth ER. This is some smooth ER that's been sliced through, and it consists of, again, this is lots of little folds and tubes made out of membrane. The smooth ER may be where things that aren't proteins get made. Cells that have to produce fats or carbohydrates may have lots of smooth ER. Your liver cells have lots of smooth ER because the smooth ER is the place where enzymes start breaking down toxins. If you drink alcohol, that is technically a poison, and your liver cells break it down, and the breakdown process happens on the smooth ER. So many proteins may get made on ribosomes that are bound to the rough ER. Incidentally, there are also ribosomes that are just floating free. They're not all bound to the ER. But proteins that are made in the ER pass through all those folds, and they end up in a body called the Golgi body. And again, that's named after the guy that discovered it, who won a Nobel Prize in 2005. And you're looking at a slice through the Golgi body right there. The simplest way to visualize one would be stack 10 deflated beach balls, one on top of the other. The Golgi is a stack of flattened membrane sacs. Looks a little bit like a stack of pancakes, except unlike pancakes, these stacks are hollow. So like right there, There's a little bend in this thing for some reason, but right there is one of those membrane sacs. And of course, there's another one up here on top of it that I'm outlining. Sorry, I'm not a great sketch artist if it's just my finger on a mouse pad, but there. So that's a cross section through one of these stacks of membrane sacs. That's a Golgi body. And you might notice there's a whole lot of little circles right up here. Those are vesicles. And there's another vesicle over here on the side, and there's another vesicle, and there's another vesicle, lots of little vesicles. Vesicles are constantly budding off the Golgi. Remember that since these membranes are two-dimensional films of liquid like soap bubbles, they can bud off and split away a structure like the Golgi and then fuse with another structure that's made out of membrane. So anything passes in the Golgi will pass from the Golgi into these small vesicles. Don't worry about the difference between, there's a lot of terms here I would not expect you to know. So don't worry about unregulated versus regulated membrane fusion. That's not relevant for our purposes. You can take cell biology in our department if you really must know. But just know for our purposes that these vesicles move from the Golgi to the outer membrane and they fuse with it and release the contents. Whatever's in those vesicles gets released to the outside. That's a process, by the way, that's called exocytosis. Cyto means a cell, exo means outside. This is the way things get outside the cell. And I would expect to ask questions on the final that might include things like, but not limited to, what type of cell would you expect would have a lot of ER and many Golgi bodies? And the answer might be some type of cell that produces lots of protein and exports it to the outside. So that's one possible source for questions. I showed you those vesicles. Those are pretty small. These happen to be plant cells. And I'm outlining here, this is one plant cell. And because it's a plant, it's got a thick cell wall outside the cell membrane made of cellulose because it's a plant. This thing right here that I'm circling is a chloroplast. You saw those when you looked at Elodia way back in the second lab. That's where photosynthesis happens. And right in the middle of the cell, there's a whole lot of empty space. That's a big fluid-filled sac. And that's called the central vacuole. Okay, an answer to the question I asked about a cell with a lot of ER and many Golgis. It would be any sort of cell that makes a lot of protein and then spits it out. So cells in your intestine that release digestive enzymes, the cells in your liver that produce the proteins that your blood needs to clot. I would not expect you to know those types of cells right away, but those might be multiple choice. This is a multiple choice question. Those might be correct answers. And I'd give you enough background information that you should be able to identify them. And the cat is losing her tiny little mind. You can't see this, but she's being very funny right now. Anyway, as for those plant central vacuoles, they contain a lot of water. And as long as they contain lots of water, they're stiff. You know, if you imagine a water balloon that's filled with lots of water, as long as it doesn't burst, it is pretty firm. And so if you have, leaves, let's say it's the lettuce in the supermarket, as long as that lettuce has lots of water in the central vacuoles of the cells, that lettuce, those cells are going to be pretty firm and the lettuce is going to be nice and crisp. If the lettuce starts drying out, these vacuoles begin to collapse. They start losing their water and the lettuce wilts, which is why if you go to Kroger, they periodically spray the lettuce with these automatic sprinklers. That's to keep the vacuoles from collapsing and the lettuce from wilting. Okay, that's actually a pretty good place to stop, because the next structure I wanted to talk about is called a mitochondrion. And you can think of these as the power plant of the cell, which was a meme recently that's kind of funny. But I promise on Monday, I will get to more detail as to what I really mean when we say that mitochondria are the power plant of the cell and why that might actually matter. But right now, it being 10:50, I will stop the recording.